Hello students, today we will discuss about the Raxi Laminas, which are actually the part of the grey matter of spinal cord. You have this question in exam, write the short note on the laminar architecture of your spinal cord, or sometimes you have the question on the cellular architecture or cytoarchitecture of the grey matter of spinal cord. So all of these are the same thing where you have to write down about the Raxi Laminas of spinal cord. So whenever you are having this word Raxi Lamina, you always keep this thing in mind that we are talking about the grey matter of spinal cord. So let's see what we have done in the previous class, that we have seen the different nuclei of the spinal cord grey matter. So let's revise them first. Now this is your green color H shape grey matter of the spinal cord, and you know that in this grey matter of spinal cord you are having the three horn. So which are these three horn? Anterior horn. Now this anterior horn is the broad area of the grey matter, and this broad area is further having the three sets of the nuclei: medial, central and lateral. So here you will have the medial, then here you will have the lateral, and then this is the central group of the nuclei. But dear students, you should know that these nuclei are further divided into the different subnuclei, which I already told you in the class of the grey matter of spinal cord. Now the second horn is the posterior horn. Now this posterior horn is having a pointed area posteriorly, and then you will have this posterior horn like this. Now this is known as the base of the posterior horn. This area is known as base of the posterior horn. This is known as apex of the posterior horn. Now in this region of the posterior horn, we have seen marginal nucleus, substantia gelatinosa, nucleus proprius, Clark's column, and visceral afferent nucleus. Now this visceral afferent nucleus is present on the base, which may present in your book. Some books will not mention, some books mention that there is a visceral afferent nucleus which is a part of posterior horn. But these four are very constant nuclei which you will find in each and every book. Now here again you have to keep this thing in mind that these nuclei which are present here from this dorsal to ventral or posterior to anterior side, you know that this is the marginal or supramarginal nucleus. Then you will have the substantia gelatinosa, then you will have the nucleus proprius, and then you will have the Clark's column, or you can say dorsal nucleus. Clear? Now you have to keep this thing in mind that some of the nuclei are present in some specific part of the spinal cord, and some of the nuclei present in the entire cord of your spinal grey matter. Now the lateral horn. Now you know that lateral horn is the laterally produced extension. Now this triangular area, which is again not a classical feature of all the areas or all the sections of a spinal cord. Now you find the lateral horn basically in the thoracolumbar area. That means it is basically having the preganglionic sympathetic neurons, and this lateral horn, we have seen that it is having the two sets of the nuclei, intermediolateral and intermediomedial group. So these are the nuclei which are present in the grey matter. But what happened that for the practical purpose we have a alternate way of the classification, and that is known as the laminar architecture of the grey matter of spinal cord. Now this is an alternative way of the presentation of the nuclei. You are having the two way of the presentation. One is which we have already done, you can write down only the name of nuclei. The second way is that rather than using the name of the nuclei, you are using the Roman number. Now when you are using the Roman number, you are denoting them from the 1 to 10 in Roman number. Clear? Rather than using the name of nuclei like substantia gelatinosa or Clark's column, we are giving some specific number like Raxi Lamina 1, Raxi Lamina 5, Raxi Lamina 10, such like. So these numbers are always used in a Roman number, and we always start giving numbering from the tip of your dorsal horn or posterior horn. That means this tip is having the number 1 and we will go from posterior to anterior side. Now, this concept is most of the time used for the experimental works only to see the termination of afferent. Now, this is what sometimes you have this question that Raxi Lamina is basically define the afferent termination. It is not used for the motor terminations. That means what are the afferents are entering into the spinal cord. So, there are two things which you have to understand, that afferents are coming from first dorsal root, and dorsal root you know that it is carrying the fibers which are the neurons which are present in the dorsal root ganglia. So, you know that these are the entry or afferents through the dorsal root of spinal now. And second is the descending tracts which are coming from the brain, and these descending tract also terminating into the spinal cord. So, one is higher centered sensations which are coming from the with the help of descending tract, and second is the dorsal root. So, these afferents, dorsal root fiber and descending tract fibers are actually the target to study such kind of the practical or you can say experimental studies by dividing this gray matter into the lamina. And you have to understand that these afferents will terminate in each and every part of the spinal cord. We know that this anterior horn is having the motor neurons like alpha, like gamma motor neurons, and we know that these neurons are going to supply your skeletal muscle. But my dear students, you have to understand that these areas also receive some terminals from the descending tract. And in the same way, you are having the lateral horn here. Now, these neurons of the lateral horn which are giving the fibers which are exit through the ventral root also receive the termination of the descending tract. And the posterior horn are also receiving the termination of descending tract. It means that these descending fibers are modifying or modulating the all sensory as well as the motor output. And how you will come to know this? You will come to know by the experiments, and for this purpose you will divide the whole area into the different laminars. Clear? So, the most important question asked in the exam, that Raxi laminar architecture is used to define, answer is termination of afferents into the spinal cord. So, now sometimes you have one more question here, who is Raxi? So, Raxi is a Sweden scientist. Raxi is a Sweden scientist. Now, you know that these are the name of nuclei which we have revised just now. Now, rather than using the name of the nuclei, I am using the 1, 2, 3, 4, 5, 6, 7, 8, 9 Roman number. So, you can see that the numbering start from 1 and it end at the number 10, but it is the symbol in Roman. Clear? Now, what do you mean by the Raxi Lamina 1? Raxi Lamina 1 denotes to the postromarginal or dorsomarginal or marginal nucleus. You know that this is the site of the marginal nucleus. So, on this side, we are dividing this spinal cord into the two equal half. On this side, I am drawing the area of nuclei, and on this side, I am drawing the Raxi lamina corresponding to the nuclei. Clear? So, this apex, you know, it is having the marginal nucleus. Now, on this side, rather than using the word marginal nucleus, I am just making a block and I am just writing here 1. Which number? 1. So, this is the Raxi lamina 1. And if I am writing Raxi lamina 1, it denotes the dorsonuclear area or nucleus of the gray matter. Clear? Now, we will talk about the Raxi lamina 2. Now, you know that there is the next number is the 2. Now, this is the area 2, and you know that which nucleus lies below the supramarginal nucleus. So, here you will have the area of substantia gelatinosa. Now, this substantia gelatinosa here denoted by the Raxi Lamina 2. So, this is again the question of your exam. Suppose you are attempting some MCQ, you will have the question Raxi Lamina 2 is occupied by which dorsal horn nuclei? Answer is substantia gelatinosa, or vice versa, that substantia gelatinosa is denoted by which lamina according to the Raxi Laminar architecture? Answer is Raxi Lamina 2. Clear? Now, you will have the next nucleus is nucleus proprius. Now, you know that in the sequence of these nuclei, you will have the third is nucleus proprius. Now, this, if you will draw the nucleus proprius, this nucleus proprius is a little bit longer. It occupies a little bit larger area because it is having more number of neurons. So, here when you will draw, it occupies generally the two lamina, lamina 3 and 4. So, I am drawing the lamina 3 and lamina 4. So, this lamina 3 and lamina 4 both are for this nucleus proprius. Clear? Now, there is some variation in the books. Now, some book says that the lamina 5 also occupied by the nucleus proprius. Some book says that lamina 5 is also occupied by the nucleus proprius, but in all the books you will find the constant number 3 and 4. So, suppose I am having this question in exam that Raxi Lamina 3 and 4 is occupied by, then the answer is nucleus proprius. The 5 is variable book by book. Now, some book says that Raxi Lamina 5 is having the base of the dorsal column. Now, where is the Raxi Lamina 5? Now, this is the Raxi Lamina 5 which occupied by the base of the dorsal column. Now, when you will see the Raxi Lamina 6, now this Raxi Lamina 6 is basically having areas which shows the enlargement. You know that there is a two enlargement, one is cervical and another is lumbosacral. So, this Raxi Lamina 6 is present in the cervical and lumbar enlargements, and you know that when you are having the sequence of the nuclei in the dorsal horn, if you start from the apex, first is the supramarginal nucleus, then substantia gelatinosa, then nucleus proprius, then what is the next? Clark's column. So, here when you are drawing the dorsal nucleus of Clark or Clark's column, now here if you will come for the laminar architecture, you will find that the 6 and 7, these both, the 6 and 7, both are occupying the area for Clark column. But here the question is again quite confusing. You have to keep in mind that 7, Raxi Lamina number 7 is having major contribution of Clark column plus your lateral horn nuclei. So, this is a constant finding in all the books. Again, this part is controversial. Some says that a small portion of Clark column is extended into the Raxi Lamina 6. So, up to the 7, what we have discussed is Raxi Lamina 1 is supramarginal nucleus, Raxi Lamina 2 substantia gelatinosa, Raxi Lamina 3 and 4 is nucleus proprius. Now, 5 and 6 are having some controversy, but if I am talking about Raxi Lamina 7, it is having the 3 nuclei, Clark's column or dorsal nucleus of Clark plus the lateral horn nuclei, that is your intermedial-medial and intermedial-lateral nucleus. So, now if you have a vice versa question that Clark column occupies which of the following lamina. Now, if you are having the option 7 only, then there is no doubt, but if you are having the option 6 and 7 both, then my suggestion is that you should take both, because it is occupying a small portion of the 6 also, but the major portion of the Clark column is defined in the Raxi Lamina 7. Now, we will move to the Raxi Lamina 8, 9 and 10. Now, first I will tell you about the Raxi Lamina 10. Now, this Raxi Lamina 10 is present around the central canal. Now, this area is surrounding the central canal, and this area is known as substantia gelatinosa centralis. What is that? Substantia gelatinosa centralis. So, here you will write the Raxi Lamina 10, and what is the area? Substantia gelatinosa centralis. Now you left with the 8 and 9. They are occupying your anterior horn. So again, this is the question of your exam, that which of the following lamina is not present in the dorsal horn. So 8 and 9 are not present in the dorsal horn. 8 and 9 present in the ventral gray matter horn. Clear? Now what do you mean by 8 and 9? Now you have to understand that there are three group of the nuclei. You know that this is the medial group, this is the lateral group, this is the central group. You know that there are three groups of the nuclei into the anterior horn: medial, central and lateral. But my dear students, you have to understand that Raxi Lamina 8 is also present in the anterior horn, but it is not talking about this medial group of the alpha gamma motor neuron. It is actually the area which surround this medial group further. So this outer area is also known as medial group, and this is also known as medial group. But this is your motor medial group and this is non-motor medial group. Clear? And this is the Raxi Lamina 8, which is a part of anterior horn, but it is a non-alpha gamma motor neuron. Clear? Now these three areas, that means the medial motor neuron, central motor neuron and lateral motor neuron, all these three are included into the Raxi Lamina 9. So my dear friends, you have to understand this laminar architecture, that when you will start from the posterior horn to the dorsal ventral horn, you will start from the 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. Clear? Now this area is, you have to mark carefully that medial part of the anterior horn is having two sets of the lamina, 8 and 9. The 9 is the motor neuron area and it is surrounded by Raxi Lamina 8. Clear? Now, when you will see the extension of these lamina, you should know this area also, because sometimes you have again this question that postero-marginal nucleus, which represent the Raxi Lamina 1, is extend in the chord. Answer is entire length. Clear? Now here you can see that I marked the red color areas which are having the extension in entire chord. So what are these? One is the supramarginal nucleus. Second is, you can see here, is the substantia gelatinosa. Then you have the nucleus proprius. Then you will have the medial group of the anterior horn, which is having non-motor area. That means Raxi Lamina 8. I am talking about Raxi Lamina 8. So it is also extend in the entire chord. Plus you will have the medial horn, medial nuclei of your anterior horn, which is also present in the entire length. But the central and lateral group of the anterior horn does not have the extension in the entire length of the spinal cord. Then lastly, the Raxi Lamina 10, it also having the extension in the entire cord. So this is the important thing to understand, that whenever you are having the question about the extension of these nuclei or lamina in the spinal cord, you have to understand that Raxi Lamina 1, that means supramarginal nucleus, Raxi Lamina 2, that means substantia gelatinosa, Raxi Lamina 3 and 4, that means nucleus proprius. Then you will have the Raxi Lamina 8, that means your non-motor part of the anterior horn which surrounding the medial group. Then the medial group of the anterior horn, that means the motor area which is the part of the Raxi Lamina 9, and the Raxi Lamina 10 is having the extension in the entire cord. So remaining are not extend in the entire cord. So if you will talk about the dorsal nucleus or the Clark's column, it is having extension only from C8 to L3 column. If you will have the lateral horn nuclei, they are extend only from L1 to L2 or L3 segment. Clear? And then when you will have the remaining nuclei of the Raxi Lamina 9, that means central group and lateral group, you will find that they are present in the cervical and lumbar enlargements. So my dear students, again I am repeating this thing, that whenever you are reading the gray matter is having the elongated columns of the nuclei, and these columns are not present in the whole length of a spinal cord. It is not a thumb rule. Some of them are present in the whole length, some of them are present in a specific part of the spinal cord. So this is again a very important question for your exam, that which of the following nuclei or Raxi Laminars are having the extension in throughout length of the spinal cord. So you have to remember only these names. Remaining are having the specific extensions in the spinal cord gray matter. Now what is this? Now suppose I am having this spinal cord. Now this is your spinal cord. You know that spinal cord ends at the lower border of L1. So this is the lower border of L1 and this is the lower end of conus medullaris. Now suppose I am drawing one of your Raxide lamina, or you can say the nuclear column. Suppose I am drawing the supramarginal. So we know that supramarginal extends in the throughout length. So here I am drawing that this is my length of your supramarginal nucleus, which is going in the whole length. So what do you mean by whole length? It starts from the first cervical segment and it ends into the coccygeal segment. So this is the coccygeal segment. So I can draw this in the whole length. Clear? And this is your cervical segments, this is your thoracic segments, this is your lumbar segment, these are sacral segment, and this is the coccygeal segment. So this is the coccygeal segment here, this is the C1 here. So now you can see that if I have to draw the Raxide lamina 1, or you can say the supramarginal nucleus, I know that it is present in the whole length, so I have drawn it. Clear? Now suppose I am having the question that what is the extension of the lateral group. Now we know that lateral horn or the lateral group of the nuclei are present from T1 to L2 or L3. So where is the T1? So this is the T1, and where is the L3? Now this is the L3. So I can draw these nuclei only from this to this segment. Clear? Because it is not present in the whole length, I cannot draw it in the complete length. In the same way, suppose you are having the question on the dorsal nucleus of Clark column. Now dorsal nucleus of Clark column starts from the C8 and it ends at the level of again L3. So I am drawing, this is your Clark's column. Clear? Now suppose you are having the question that which is the extension of medial group. Now you know that medial group of the anterior horn is having the extension in entire cord. So again I can draw, this is the medial group which is having the extension in entire cord. So again you have to draw it in the anterior length of the spinal cord gray matter. So in this way you have to remember that whenever you are having the extension of spinal cord from C1 to coccyx, you have to keep this thing in mind that every nucleus is not having the extension from top to bottom. Some are present only in the some specific segments of the gray matter of spinal cord. So I hope it is very clear to you now. Now we'll move to the functions of these Raxide lamina. Now when you will do this architecture division and when you will have the experiment, what is the output? So output is this, that when we have the Raxide lamina 1 to 4, we find out that these Raxide lamina which are present in the dorsal horn are actually the point where they receive the cutaneous sensation. And you know that the cutaneous sensation or the afferents which are entering into the spinal cord through the dorsal root coming from the first order neuron, and these neurons are present into the dorsal root ganglia. And apart from that, these are also receiving collaterals from the ascending tracts of the dorsal funiculi. They are also receiving some ascending tract collaterals. The tracts which are going upward also giving some kind of the collaterals. So what is the function of these Raxide laminas? The function is that they give rise to a spinothalamic tract fiber. So what is a spinothalamic tract? It is a ascending tract, and this tract carries the pain and temperature. So we will find out that which type of cutaneous sensation relay in Raxi Lamina 1 to 4. Answer is pain and temperature, and they will give rise to the spinothalamic tract fiber. The next group comes is your 5 and 6. Now I told you that 5 and 6 laminas are quite controversial as if you will go through the different books. But the common thing which will come out from the experiment of the Raxid, that these 5 and 6 laminas regulation of the pattern of the movement by process of the proprioceptive information from the muscle. That means they receive the proprioception from the dorsal root. So this is the question of your exam, that Raxid lamina 5 and 6 are related to which type of the senses? Answer is proprioception. So what is the output? Now they are receiving the proprioception, and these proprioception are processed by the fibers of the corticospinal, that is a descending pyramidal tract, and rubrospinal tract, which modulate, or the processing of these proprioception will take place, and ultimately these movements are of the muscle. The processing of the proprioception is helpful in the movement or the pattern of the muscle movement formation. Clear? So regulation of the pattern of the movement by processing of the proprioception information of the muscle is helpful to find out the pattern of the movements of the muscle. Clear? So we have seen that Raxid lamina 1 to 4 are basically related to the pain and temperature. Raxid lamina 5 and 6 are related with the proprioceptive impulses from the muscles. Then Raxid lamina 7. Now this Raxid lamina 7 is connected with the cerebellum and the cerebrum. And I told you that Raxid lamina 7 is having 3 nuclei. One is the dorsal nucleus of Clark. Second thing is that they are having the nuclei of your lateral horn. So these lateral horn nuclei are actually autonomic preganglionic fibers. So these two nuclear group which are present in Raxid lamina 7 are important to give rise to the spinocerebellar tract. Now this spinocerebellar tract is not arising from both the nuclei. This spinocerebellar tract arises from the Clark's column or dorsal nucleus of Clark. What I mean to say? I mean to say that the Raxid lamina 7 is having two sets of the nuclei. Nuclei of the Clark column and lateral horn nuclei. But when you will see the function, the Raxid lamina 7 give rise to the spinocerebellar tract, and these fibers of spinocerebellar tract are the fibers of which nuclei? Dorsal nuclei or anterior this lateral horn nuclei? Answer is dorsal nucleus of Clark column. So dorsal nucleus of Clark column, which is a part of the Raxid lamina 7, give rise to the spinocerebellar tract. But there is one more important thing, is that they accommodate the autonomic nervous system neurons, which is a part of the lateral horn nuclei, which are intermediomedial and intermediolateral. Then we will talk about the Raxid lamina 8. Now where you will find Raxid lamina 8? I told you that it is present in the anterior horn, and this area surrounding the medial motor neurons. So here you will find that this Raxid lamina 8 receives the descending terminals from the vestibulospinal and reticulospinal tract. Now you know what is vestibulospinal? Now you know that vestibular apparatus is related with the balancing. So they are receiving the vestibulospinal fibers and reticulospinal fibers. So what will happen, that these input of the your Raxid lamina 8 will go into the Raxid lamina 9, and what will happen ultimately, they will modify the output of motor neurons which are present in the Raxid lamina 9. Clear? What do you mean? Again I will explain this, that this is your anterior horn. You know that anterior horn is broad. In anterior horn, this is the medial part where you will have the Raxid lamina 9. Clear? This is the central part, this is the lateral part. Now this 9 is surrounded by the 8. Now this 8 is receiving the fibers from the vestibulospinal and reticulospinal tract. Now, these fibers of the descending tract will project into the Raxi Lamina 8, and then the fibers of the Raxi Lamina 8 will modulate the output from this medial motor neuron of Raxi Lamina 9. Clear? So, you have to first keep this thing in mind that alpha motor neurons which are present in the anterior horn of the grey matter is having multiple influences, and after those influences the output will come out. Clear? It is not like that whatever the alpha motor neurons are sending the efferent impulses, they completely 100% come out. No. Whatever the output generated by the alpha motor neuron to approach the skeletal muscle, first these outputs has been modulated by n number of the influences. So, we have seen one of the influences that vestibulospinal and reticulospinal tract. Clear? Then we will talk about the Raxi Lamina 9. Now, Raxi Lamina 9, we have seen that it is having the three major set of the alpha gamma motor neuron. That means the medial motor neuron group, central motor neuron group and lateral motor neuron group. But again, I just told you that whatever the alpha motor neurons and gamma motor neurons are there, their output is not 100% approach to the skeletal muscle. That output is first modified, and the modification is done both by the sense of ascending and descending tract. So, the descending tract which are modifying the output of alpha and gamma motor neurons are corticospinal, rubrospinal, vestibulospinal and reticulospinal. Clear? So, this question has been asked so many times that alpha motor neurons is also known as final output pathway. It is also known as final output pathway of your spinal cord. Why final output pathway? Because whatever the modification is taking place in the output of alpha motor neuron will take place in the anterior horn, and these influences are coming both from ascending and descending tract. So, these are the descending tracts, that means corticospinal, rubrospinal, vestibulospinal and reticulospinal. Those are giving their collaterals, and these collaterals will go and modify the output of alpha motor neuron. Clear? Now, what is the function? What is the difference between the function of alpha and gamma? So, alpha motor neuron basically supply the extrafusal fibers of skeletal muscle, while gamma motor neuron supplies the intrafusal fibers of the muscle spindle. Clear? So, this question has been asked so many times that alpha motor neuron supply which part? Answer is extrafusal, and gamma motor neuron supply which part? Answer is intrafusal fibers of muscle spindle. Clear? So, now at the end of this session, what we are able to understand? We are able to understand that what is the Raxi Laminar architecture of grey matter, how you are correlating the Raxi Lamina with the different nuclei present in the grey matter. The another important thing which you have to understand that all the nuclei, or I should say the Raxi Laminas, are not extending in the entire length of the cord, and the most important thing is what are the different functions of these Raxi Laminae. So, this is all for the today's session. Thank you.